In this session, let us study about nucleosomes and higher order chromatin organization. Let us begin with the nucleosome model. The nucleosome model of chromosome is a scientific model which will explain about the organization of DNA and the association of DNA with proteins in a chromosome. It also explains about the exact mechanism of how the DNA is folded in the nucleus. And this model was proposed by Roger Kornberg in 1974, and it is one of the most accepted model of chromatin organization. This model was confirmed and christened by P. Audit and others. Let us know what is a nucleosome. A nucleosome is a complex of two things. One is the DNA and the other is the proteins. Here the proteins are histones. The DNA will be wrapped around these histone proteins. It forms a complex called chromatin, and the DNA has to be condensed into a smaller volume. This will give the chromatin a beaded-like appearance on a string. So this is the chromosome in which this part is enlarged. Here we can make out that there is the DNA which is wrapped around these histone proteins, and this single unit here is called as a nucleosome. The very long DNA molecules have to be packed in the nucleus in about a few micrometers in diameter, so it has to be done in an orderly manner, and here the histones will play a crucial role in packing this very long DNA. Thus, these nucleosomes are called as the fundamental packing unit particles of chromatin. Let us understand the concept with an example. In this picture, you can visualize the Maggi noodles, which is uncooked, and this is cooked. The uncooked product here is arranged in an orderly manner and it is condensed, whereas the cooked product occupies a large volume. In the same way, the cell contains the nucleus inside which there are chromosomes which are compactly arranged in this way. So when this chromosome, when it is stretched, it forms a chromatin thread along which there is the beaded-like structures which are called as histones. These are the beaded-like structures, and it is enlarged view here in which you can see these are the histones and this blue structure is the DNA. The DNA always wraps around the histone proteins, which are the core proteins, and it is always in the ratio of 1:1. These are the core proteins, which are histones, around which DNA is found to be wrapped. The histones are low molecular weight proteins and they have large proportion of positively charged amino acids. So the proteins are positively charged here and the DNA is negatively charged. So these two, the proteins and the DNA, are held together because of these charges. There are 5 types of histones. They are H2A, H2B, H3, H4 and H1 linker histone. This one is the H1 linker histone and the others are H2A, H2B, H3 and H4. Out of all of these, the H2A, H2B, H3 and H4, they are the nucleosomal proteins or the core proteins. And these are 2 molecules of each and they form an octamer of nucleosome. You can see that there are repeated units, 2 units of H2A. This is H2A, these two. This one is H2B. This is also H2B. And this is H4. This is also H4. And this in orange is H3. and this is H1, and the purple one is the DNA helix. Here the nucleosome is made up of 2 H2A, 2 H2B, 2 H3 and 2 H4, whereas H1 is present only once and it is loosely bound with the DNA. These nucleosome beads are separated by a linker DNA, and this is about 54 base pair long. It contains a single H1 histone protein molecule. The DNA is found to be wrapped twice, that is, it completes 2 turns around the histone octamer. It is about 200 base pairs. After understanding the concept of nucleosomes, let us understand the higher order chromatin organization. In human beings, there are 46 chromosomes, and the DNA content of all these chromosomes, if they are laid out, it would measure about 2 meters and the diameter would be only 2 nanometer. So this DNA must be tightly packed to fill the cell's nucleus. At the same time, they should also be available for expression of genes. So how is this done? There are a number of ways in which the chromosomes can be compacted or arranged compactly. The chromatin is organized in 3 levels. In the first level of packing, the DNA is wrapped around the protein and it produces a beaded-like structure called nucleosome. This is the DNA part which is enlarged, and this DNA is about 2 nanometer in diameter. And the beads on a string, they appear to be around 11 nanometer. The second level of packing, in this level we can see that there is coiling of beads in a helical structure. This appears to be around 30 nanometer and it is seen during interphase chromatin and in the mitotic chromosomes. These are called as solenoid structures, and this part is enlarged and shown here. Here this region contains about 6 nucleosomes per turn, and this consists of the nucleosomes which will be around 300 nanometer. And in the final level, the fiber is found to be organized in the form of loops or the scaffolds and domains, and this is about 700 nanometer in size. It is seen in the metaphase chromosome, and there is an extensive looping of DNA at this stage. This is a picture which depicts all the 3 stages of the chromatin organization. This is the DNA, these are the nucleosomes which form the first level. This is the solenoid structure which is the second level of organization, and this loop structure is the final level of organization of chromosome. So this is all about the nucleosomes and higher order chromatin organization. Thank you.